Here at ESGO, we present some interim analysis of the PYNNACLE trial, and we have selected different tumor types who had this specific mutation. And these patients were treated with Rezatapopt, which is this drug who, let's say, targets the undruggable target.
P53 is an oncosuppressor gene and it is mutated in the vast majority of ovarian cancer patients. For several years it has represented a very interesting target, but unfortunately we never succeed in targeting P53 mutation until today. We are in ESGO and Professor Frenel just presented the results of the PYNNACLE trial with a new drug targeting a specific mutation of P53. Jean-Sebastien, can you tell us about your drug and your trial?
Yes, so the PYNNACLE trial is a basket trial which has included patients harboring this specific mutation, the TP53 Y220C mutation. So these patients were treated with the Rezatapopt, which is an oral drug which targets specifically this mutation. And what we present here today, this is an interim analysis of these trials regarding the overall response rate for ovarian cancer patients. We have included 51 patients with ovarian cancer, and these patients had mainly platinum-resistant disease, some patients had platinum-refractory disease. The objective of this trial is to demonstrate the overall response, which is centrally reviewed. This is the main objective. And the second objective are PFS, of course, and overall response rate as well with the investigators. Out of these 51 patients, we had 48 patients with evaluable disease. So we have demonstrated a high overall response rate, which is around 46%, which is very important compared to our traditional comparators. What we learn again from this trial is that the response arrives very quickly because the median time to response is 1.3 months, which is very short. And the duration of response, which matters a lot for our patient, is 8 months. So on our side, it's a very important and very interesting result for this trial.
For sure. I was totally fascinated by your data, and I was totally fascinated by the mechanism of action of this oral drug, because it seems that this particular mutation creates a kind of task in the protein, and this drug is able to link there and stabilize the wild type version of the protein, which is totally fascinating to me. What about the toxicity profile of this drug?
So, this drug is an oral drug, and treatment-related adverse events in this interim analysis were very, very manageable. So mainly when grade 1 and 2, some nausea, some fatigues, some laboratory abnormalities were very manageable, with only one patient who stopped the drugs for treatment-related adverse events. So the impression is very good in terms of tolerability for this drug. So that's where we are. So I'm curious about what do you think about this?
Oh, I have to say I was very impressed by the results. To see for the first time this possibility for our patient it's incredible to me. It's a rare mutation, but represents about 1% of all solid tumor. It's more higher in ovarian cancer. It's about 3% and even higher in high-grade serous tumor that represent the vast majority of ovarian cancer. So this is a possibility for our patient. But we need to know and to search this patient. And it is mandatory now that we have this opportunity to search this patient systematically with an NGS on tissue with a wide coverage for P53 mutation. For sure, there is the alternative of liquid biopsy, but right now the data on liquid biopsy are not so strong, so I would suggest to reserve liquid biopsy just in situation in which tissue is not available, providing that the liquid biopsies, the circulating tumor DNA, has excluded the chip. This means another important aspect that we need to have a closer collaboration with our molecular pathologists that should identify this patient as soon as possible in their clinical pathway.
Thank you.